It's a pleasure to be here, and thanks to the Larrys for the invitation. I did speak here 5 years ago, but it was via Zoom, and so I was always keen to come back. My friends have told me what a marvelous time this is, and they did not exaggerate. I made the mistake of putting my sweater on after you mic'd me up. I was afraid that might happen. All right, so this is my current lab, and I should say that I'm going to give a very different talk than Jack did. I'm going to talk only a tiny bit about our own work, and I'm going to try to give you a survey of why you might be interested in this field of circadian rhythms. But the work I'm going to talk about from my lab is done by Nick Peterson. I should say that Nick, in addition to his scientific mettle, also has a very good sense of humor. He was raised as a Mormon, which is relevant to the little anecdote I'm going to tell here. So there was some food trucks outside of Brandeis, and somebody pulled up a truck next to the food trucks, and on the back of the truck there was a guy giving out pamphlets. The side of the truck said "Jews for Jesus," and Nick went up to the guy handing out the pamphlets and said, "Excuse me, what's the exchange rate?" And so we have this, the school, Brandeis has this origin, a particular origin by the American Jewish Committee. And I should say that the work I did on circadian rhythms was done in collaboration with my colleague, Jeff Hall, with whom I won this prize along with Mike Young from Rockefeller. And Jeff and I bonded from the day I arrived at Brandeis when I was 30 years old. I should say, a shared sense of tragedy. You know, they say the Jews experience every tragedy twice. First they imagine it's going to happen, and then if you wait long enough, it happens. And so, and then of course, Jeff, being of Irish origin, had really the great English poet Yeats in his background, which applies to my ethnic group as well as the Irish. Namely, we have an abiding sense of tragedy that sustains us through brief periods of joy. A more poetic description than the one I gave. All right, so let me begin by recapturing something that Jack talked about, namely the origin of life, and in this case, the fact that early life evolved by being exposed to this inexorable light-dark cycle. And this is way before the atmosphere was oxygenated. It was a reducing atmosphere. Nutrition was nothing like what currently exists on the planet. And this is really the oldest Zeitgeber, or environmental cue, that life ever had. And as a consequence, and I would argue as indication of how important clocks are, they've really evolved differently in different kingdoms, or at least this is the current way things look. That is, bacterial clocks, plants clocks, and animal clocks are really quite different from each other, and I'm going to talk about animal clocks. So let me just give you some definitions so that you can get on the same page. Circadian rhythms are self-sustained. They keep ticking in darkness. They have a period that is not exactly 24 hours. They're circa 24 hours, about 24 hours, 24 hours and 15 minutes in humans. And they get to be 24 hours. We get to be exactly 24 hours, except for a few of us who are perpetually late, by entraining to the light-dark cycle, which of course is exactly 24 hours. So our slightly slow clocks are reset every day by light, and that puts us back on schedule. The purpose of the clocks is anticipation. The early bird gets the worm, and the early worm avoids being eaten by the bird. And there's some internal processes that are also sort of kept in sync. They work better in series than in parallel, et cetera. So that's the idea. Now, I very much like this quote from Sidney Brenner. He appeared, of course, in Ritchie's. We go back 60 years, and he was Ritchie when I first met him, and I was Mike, of course. So anyway, the old nomenclature dies hard. So this is a quote from Brenner that I'm very fond of. Progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. Ideas are cheap in biology. Most of them aren't the way evolution actually works. New techniques, of course, it's an oversimplification because to devise a new technique, you have to have a new idea and so forth and so on. So, and discoveries are the things you stumble across and you say, gee, I wonder why that came about like that, and of course you try to track it down. So I want to make the argument that the assay of circadian rhythms, namely locomotor activity rhythms, is just a remarkable discovery. And the fact is that every single animal is almost identical to every other animal. And this is unprecedented in behavioral biology. And it means, of course, you can do genetic screens, because the outliers are several standard deviations off the norm, because every single wild-type animal from a particular species is exactly the same as every other animal. And so this is a wheel-running record. In a light dark cycle, it's kept on track, and then, of course, it drifts, in this case, slightly less than 24 hours in constant darkness, DD, and that drift indicates the endogenous period, and every animal is exactly the same as everyone else. So we started doing this in flies, following in the footsteps of a pioneer, which I'll mention in a moment. And the way we do this is to have individual flies in tubes. They run back and forth. There's monitoring, a beam break device, and we get the same kind of wheel-running records with the flies that you would get with mice or rats, or for example. And these are records of flies and mice that are side by side, and just gives you a picture. One is, of course, the mice are shorter than 24 hours. The flies are slightly longer than 24 hours, but it's the same general picture, and you generate the endogenous period in the same exact way. So genetics, the mantra for genetics is phenotype, just like location for real estate. You have a great phenotype, you can do a screen. That's the idea. And of course, the beginning of this whole era, the pioneers I was referring to are Konopka and Benzer. Remarkably, this is the same Seymour Benzer that you heard about from Rich, who did the R2 work. And when he moved to Caltech in the 60s, he decided to take the same deep genetic approach to behavioral genetics in the fly and isolate mutants. And Konopka was his student, and Konopka actually brought, a 19-year-old graduate student, brought the idea of doing this screen. Konopka collected butterflies, knew that insects had circadian rhythms, said to Seymour this would be a great thing to do, and they did this. This paper, which is probably the most important paper in behavioral genetics, had 10 citations in the first decade after it was published. A lesson for those of you who chase high-profile publications. And so I'm going to say very little about the brain and the nervous system in this context. It's actually, much of my lab works on that problem, but I think it's more parochial and good for a neuroscience audience, and less for this broad audience. I'll simply say that if you take clock genes or their proteins and you use them as reagents to then illuminate the mammalian brain on the left, that's actually an in situ hybridization experiment with a probe for a clock gene across a time course. And there's a little region of the hypothalamus which lights up like gangbusters, only at sort of certain times a day. So the transcript undergoes dramatic circadian oscillations. That's the suprachiasmatic nucleus, little region of the hypothalamus. That's the master clock in the brain. It's got high levels of the proteins, and it really very robustly does this thing. And when we do exactly the same kind of assay in the fly brain on your right, we get about 200 cells that light up. Those are lit up in orange there with an antibody. And same general principle, high expression of these things, very intense, robust oscillations. And both of those two regions are the master governor of behavior, of the sleep-wake cycle in humans, of the sleep-wake cycle, locomotor activity rhythms in flies, et cetera. So I'll simply say that over a period of about 15 years, starting in flies and then incorporating the same general principles from mice led by Takahashi and his colleagues in the latter case, a very small group of proteins generate a transcriptional feedback loop, a negative feedback loop. Most of those proteins are conserved between flies and mammals, suggesting common origin, inherited from the last common ancestor, maybe 550 million years ago or so, between flies and mammals. And they both have the same feedback loop. It takes 24 hours to go around the cycle. The negative regulators are made by the positive transcription factors. The negative regulators, the two proteins at the top there in blue, period and timeless in flies and period and cryptochrome in mammals, they turn off their own transcription, and that's the cycle. There's an important kinase that also modifies these proteins, CK1, in mammals, and it's ortholog, the same exact protein in flies called double time, and I'll talk about that a little bit later at the end. So to bridge to all of you healthcare professionals, people interested in this, I want to talk for a few minutes about the impact of circadian rhythms on physiology and metabolism, human health, et cetera, these various, this list of phenomena on which clocks impact. So first question, why does the clock have such a profound influence on physiology or metabolism? And there's two simple answers to the question. The first is that this little machine functions in all the tissues of your body. It's the same, I emphasize the brain as governing behavior, but metabolism, this machine works in your liver, kidney, spleen, lung, skin, et cetera, and the same little clock is ticking away, and so that's half of the answer of why so much physiology is governed by the clock. And the other reason is that this little machine sits at the top of a molecular hierarchy within each of these cells and then governs from hundreds to thousands of output genes, which themselves cycle. So this is not part of the timekeeping mechanism itself. They're downstream, in some cases 2 or 3 layers downstream. But these transcripts, if you simply take the liver and isolate messenger RNA at different times of day, you get hundreds, perhaps up to 1,000 of messenger RNAs, which go like this, right, all the time. And if you wipe out the clock, either in the entire animal or locally in the liver, not all that goes away. So if that's happening in every tissue, it turns out that the estimates are greater than 70% of our genome is under circadian control somewhere. Right? So of course, what is cycling in the liver is different from the kidney and the spleen and the brain, et cetera, but really a large fraction, the majority of our genome is under circadian control. So it's no wonder that so much of physiology is impacted by the clock. Okay, so let me get to a few sort of subtopics which are in the wind, sort of in popular culture, and just give you a sort of walk through this so that you can then go investigate if any of them are of interest to you, right? So first of all, chronopharmacology or chemotherapy at different times of day, for instance. So it turns out that this has become of more and more interest and it really seems to make a big difference in some cases. I should say, for the cognoscenti, because it matters at different times of day doesn't really mean it's circadian, et cetera, et cetera. But, it starts to become of interest. So it turns out that immunotherapy, checkpoint stuff, works much better in the morning and than in the evening. And it turns out that what really matters is your first dose, the first injection. And exactly physiologically why that's the case is not known, but of course it's of great interest. A second area that's being worked on by some of my circadian colleagues is glioblastoma. So it turns out you can ask the question, if you have a tumor, if you wipe out the clock in the tumor, if you inhibit the clock, chemical inhibition, let's say a molecule which binds to one of the key transcription factors and inhibits them, and some of them have orthostatic pockets, and so you can use traditional medicinal chemistry to try to find inhibition. If you do that, with some cancers, glioblastoma in particular, it's quite effective. And so, and I should also add parenthetically that the frontline alkylating agents which are used for glioblastoma are also more effective at one time of day than another. And I should say there we have at least a plausible explanation, because DNA repair systems are themselves very potently under circadian regulation, right? Which makes a great deal of sense, right? Because we're exposed to sunlight at different times of day, et cetera. So this is really an area of active investigation and of intense interest how to improve this in the cases where it seems to be effective. So timing of food intake and metabolism. I'm going to harken back to an experiment that was done about 25 years ago in two different labs, Mike Menaker's lab and Uli Schibser's lab, and it's a super simple experiment, as much of biology is often illuminated by very simple things. Normally, all the clocks in our body, these diverse clocks, and you might have asked me, how do they all keep in sync, right? And it turns out that the HPA axis downstream for the brain, it causes hormones. Cortisol, for instance, is the great example. Cortisol cycles in our serum, high in the morning, get excited, low in the evening. A lot of the clocks have glucocorticoid receptors, and so the cycling cortisol shifts these peripheral clocks and keeps everything in sync. Okay, so all good, everything works together. However, if you feed a mouse at the opposite time of day during its sleeping time, so you only give it food in the daytime, of course, which mice are nocturnal, then it turns out the liver clock shifts all of its clock gene expression by 12 hours, right? So you end up having internal desynchrony between the brain clock, which is governed by light, and your eyes, and your peripheral clocks, because the food metabolites are all part of this entraining system. And of course, this lies underneath jet lag a bit, because of course we go to a new place, and you get internal desynchrony depending on how long it takes for the clocks to readjust, and so forth and so on. So you have this dramatic change. Feeding time has a dramatic change. And I should say there's no effect of this feeding change on the suprachiasmatic nucleus, as I indicated, because it's governed by its connection with the retina and the eye by light. And so here's a sort of early paper which has a surprising effect, and it's a paper that dealt with Huntington's disease of all things, and it's very simple. They took a Huntington's mouse and they put it on a feeding schedule, time-restricted feeding. feeding in its active phase, zero food in its inactive phase. And of course, mice are generally fed ad lib. You just put the food in the cage and you forget about it, right? So it turns out this actually improves the prognosis of Huntington's prognosis and the progression. It's an aging malaprop. I probably have made about 7 so far. It improves the progression of Huntington's here. And of course, this is a classic experiment done by Sachin Panda's lab, and it's been done in other places at about the same time. It's very simple. If you time restrict feed mice, and this means, for example, food only during the active phase, no food during the inactive phase, and you put the mice on a high-fat diet, FA is ad lib fed, FT is time-restricted feeding, you get this enormous difference in obesity, and the mice consume exactly the same number of calories. There's no the metabolic rate's the same, no change in respiration. So a mystery, right, scientifically, but a very clear effect that's been reproduced in several different places, and so for humans, of course, this is called time-restricted eating, and there's evidence that this has positive health outcomes, and there's a very simple rational explanation for this. Our peripheral tissues, liver as a very good example, go through dramatic time of day differences in metabolism, right? So the liver is involved in glycolysis during the day when we eat and take in carbohydrates, and gluconeogenesis at night. So all kinds of enzyme systems change, and of course the liver also, just to another example, upregulates all kinds of detoxification systems prior to our eating, our mealtime, and it's entrained by when we eat regularly. And of course, food not only contains nutrients, it contains toxins, and so the liver's doing both jobs. And so, not eating at the wrong time, snacking in the evening, et cetera, is the rationale for sort of keeping in harmony with your body's own natural cycles, not giving it food when it's not prepared for the 11 o'clock at night, the piece of pie in the fridge, et cetera. It's probably not a good idea. It makes sense. A lot of this is anecdotal, but it sort of fits with the general idea that all kinds of things are undergoing cycles. And so I'm going to segue to another topic related to, I'm paying exquisite attention to the time, young man. I told him I would quit at 35 minutes, even in the middle of a sentence. So if you suddenly hear me go silent, that's the reason. So understanding the basic science of sleep and improving human health. So I'm not going to tell this story about narcolepsy and orexin. It's a fantastic scientific story. It goes back about 25 years from Yanagisawa and Minyo, who isolated the cells, who identified the neuropeptide and the receptors. This is wake promoting. This is the system that keeps us continuously awake during the day. Narcoleptics are missing this stuff, and they fall asleep all the time. So sleep. So we understand quite a bit about the circadian control of sleep. And it turns out that, of course, we have this metronome that's ticking away, mostly in the brain as far as sleep is concerned. And it is really an alertness system during the daytime, during waking, and then it dissipates at night, so we can fall asleep. And it turns out that there are human Mendelian variants of our famous clock genes. These are the human orthologs, and they turn out to underlie human sleep syndromes. So families which have mutations in these clock genes, the human orthologs thereof, they turn out to fall asleep. They can't stay awake past 7 p.m., for example, in the case of advanced sleep phase syndrome, or you can't fall asleep till 3 or 4 in the morning, and that those are legitimate mutations in these genes which time sleep. And of course, this segues to the general issue of early birds, night owls, and chronotype, right? Are you an early bird? Are you a night owl? And chronotype, not surprisingly, is heritable, like everything else, right? And clock genes contribute to this phenotype, whether you're an early bird or a night owl. It's not exclusively. There's other alleles, there's other genes which contribute, but clock genes understandably make a big contribution to this. And here's a very sort of, I think, a very interesting, simple assay that's illuminating from the night owl, early bird point of view. The phenomenon is called social jet lag, and the idea is shown, is very simple on the left. There's a sleep schedule, and the barber pole 5 stripes are work days, and the 2 green lines underneath are free days, the weekend, right? And so this is the time you go to sleep and the time people wake up. And of course, during the workday, the alarm clock is waking people up at a very regular time so they can go to work or school. And you can see the disconnect between when they go to sleep and when they wake up on free days, because of course they're sleep deprived from the week. Every day they're not getting quite enough sleep because the alarm's waking them up after 6 hours instead of 7 or 8, and consequently they sleep much more and later on the free days. And if you look at the difference between work days and free days, you get this metric called social jet lag. And of course, night owls are much more plagued by this than early birds, because their work schedule is much more in sync with their natural rhythms, the early birds, than the night owls. And you can measure chronotype very successfully, actually, by self-reporting. But of course, you can check this with body temperature rhythms or melatonin rhythms in the people, and it works very well. In other words, the melatonin rhythms, hormone of the night, that oscillation is shifted in night owls and early birds and so forth and so on, and you get a real sort of strong picture of this. And this fits everybody's experience. It turns out that young people have later chronotypes than adults and their parents. They simply can't go to sleep as early, et cetera, with all the implications for school start time. But this is a real phenomenon. It's not just all due to screen time and drugs and stuff like that, right? Okay. Here's something that's very much in discussion in the circadian community, and I'm just simply going to sort of breeze through it to give you the flavor for it. So it turns out, and this is really legit, daytime morning light is good for you, and nighttime evening light is bad. And the evidence for this is quite remarkable. One piece of evidence is if you look on the western and eastern edge of time zones, at let's say the eastern time zone, which is 1,000 miles wide, the incidence of disease in Michigan versus Maine is quite remarkable, much higher in Michigan than in Maine, because they get an hour later sunrise, right? More light at night, less night in the morning. And this has been done in Russia and in a couple of other locations. So it's a general time zone phenomenon. And of course, this impacts daylight savings time versus standard time, et cetera. And I should say that the community is absolutely berserk, bananas, over the plans to illuminate the planet with satellites in the night to have persistent sunlight, of course, because, and humans can deal with this. We put a mask on at night and sleep, et cetera. But for all of life on the planet, which is very dependent on this light-dark cycle. It's really a very bad idea, from plants to humans. Okay, so homeostatic control of sleep. So I mentioned sleep drive, circadian drive, but of course I have to point out that there's a whole second system, which is the homeostatic control of sleep. So what's that? How much sleep we've had, how much we need. If you stay up late for one reason or another, but you have to get up in the morning, you're sleep deprived, you fall asleep at the lecture at 4 in the afternoon because you haven't had enough sleep, et cetera, and then you make up that excess sleep the next night by sleeping both longer and more deeply. And that's sleep drive. That's homeostatic control of sleep. We don't understand this. Where and what is keeping track of sleep need, right? Where in the body? What molecules are doing this? And really, of course, this overlaps with the question of why do we sleep? What is the conserved deep function of sleep? It's not that there aren't proposals, there are, but there's no well-agreed consensus, if you will, analogous to how circadian clocks operate, which has reached a consensus. There's very little outliers. So I should say there's a lot of human research on this, and I think the most exciting work out there, very much on the frontier, are sets of families studied by Louis Patrick and Fu at UCSF, which are short sleepers. It's clearly inherited in the family. They sleep legitimately 3 or 4 hours a night. Many people false report all this stuff, but you put them in the lab, you monitor with EEG. This is legit, and the report is, you would think that they suffer for one reason or another, right? This isn't good for you. They turn out for some reason to be, or so they say, to be protected from disease. They seem to be particularly resilient. They're long-lived. There's low incidence of cancer, et cetera. So the notion is that they produce extra stuff more avidly. That the restorative process of sleep is working more profoundly in them than it does in most of us. That would really be remarkable. Can we find a magic elixir to allow us to sleep 3 hours and be fully restored? And I should say that flies really sleep like we do. And so my flies, as they have done for circadian biology, illuminate this question of how and why we sleep. And I should just say that they have homeostatic control of sleep. If you sleep deprive the fly, then they sleep longer and more deeply, unambiguously. And young flies sleep more than mature flies, just like our babies do. And most interest to me, of course, aging causes sleep fragmentation in old flies. So sleep fragmentation, you wake up at 3, 4, 5 in the morning. You can't maintain sleep as well when you're old, and that's exactly the same for the old flies. So every sort of interesting human aspect of takes place in the flies. A forefront here: hospitals, lighting, sleep, food. Should hospitals change their practices? It's probable that taking your vital signs at night and waking you up is probably worse for you, interrupting your sleep, than the benefits you get from knowing those vital signs. And here's a sort of recent paper from John Hognish, my circadian colleague. And if you have a child that's getting parenteral feeding, they generally feed all around the clock, right? And because the nutrition is being supplied. And if you simply supply the nutrition only in the daytime than the nighttime, then they get out of the ICU faster by virtue of doing this kind of thing. So the lighting in the hospital, sleep, food, et cetera, all that is sort of a frontier. Damn it. Okay, I'm out of time because I really said I would stop on time. Is that correct?
No, you still have 9 minutes.
I think it was okay. I'm not going to correct him. It says I have 9 minutes. Okay, so let me segue to this last little bit and just tell you a little story from my lab. So it is the question of what determines the period. So, you have this feedback loop, but, you need to do in vitro experiments or something to really get kinetics. It's a sketch, but it doesn't give you mechanistic insight. And most interestingly, from this point of view, increases cell cycle, gets faster, all kinds, everything you choose to measure. Circadian's exception. And of course, the rationale is that this is a time, you don't want the timekeeper to screw up if a cold front, comes through Boston, and the flies should still know when the predator's mates and food is going to be around at the right time, et cetera, right? And it turns out that this is even true for mammals, because if you take a mammalian tissue culture cell, you put a reporter gene in it, okay, so you've now gotten rid of the hypothalamus, which keeps us plus or minus 1 degree C, and you do this experiment, you put the tissue culture at 2 temperatures, there's no change in the circadian period. So it's probably an intrinsic feature of the system. Okay, so the clock protein period undergoes this slow and massive phosphorylation over many hours before undergoing degradation, and this is true in mammals as well as flies. And we know that this is due to the phosphorylation by CK1, by this kinase I told you about. And it turns out that although PER is mostly unstructured, it has 2 small helices that are conserved between flies and mammals. And these 2 helices, and here we come, the miracle of AlphaFold, they dock onto CK1 with a very high probability coefficient. So the substrate, which is being phosphorylated by this enzyme, the circadian protein, not only, and it's phosphorylated all over the place, it has this binding site, and we now know something structurally about the binding site. So here's the experiment: make mutations based on the predicted structure, and then assay them phenotypically in the fly. So this is the only this is a contemporary experiment, not published yet from the lab. So here's a salt bridge that holds those 2 helices together so that it can dock onto the protein. At least that's onto the kinase. That's at least the proposal. And now it turns out if you mutate that acidic residue to a basic residue, so you have 2 lysines opposite each other here, you get this remarkable increase in circadian period, right? 1 mutation, put it in the fly, and you get this big change in period. Now, if you make a second mutation, so you change that lysine 789 to an acidic amino acid, so now basically you've restored the salt bridge, but you've changed the location. If you've changed the location, you restore the period of the animal, and of course you've also restored the temperature insensitivity, right? Because the period goes up with temperature in the mutant, and now this temperature insensitivity returns. All right, and we've done biochemical assays as well as assays in the fly. In other words, looking at the kinase function in vitro. So I wanted to do this because here I am. Tom is chairing the session, and of course I did this for a living 30 years ago. That is, I changed RNA helices, and you could show that a stem was dependent upon the base pairing, but it didn't care really whether that was an AU or a UA or a GC, et cetera, et cetera, et cetera. So it's exactly the same, and many things happen in my lab which other people, complete. So I said, why don't we do this? I know about this stuff, it should work, right? And it did. And with that, I'm going to close, and thank you very much for your attention.
Well, thank you, Michael, for giving a scientific basis for stuff that we've all observed in our everyday life and in the middle of the night as well.
You do the calling on the questions. Yeah.
Please.
Thank you. You talked about social jet lag. What about real jet lag? What can we do?
Suffer like I do. Take Ambien on the plane, to sleep. Suck it up. Everybody, it's of course extremely interesting problem, both what can I say, phenotypically and scientifically, but people working on it, but nobody has a good. Mice are very light sensitive, and it's pretty good to phase shift a mouse by putting him in front of a bank of lights before the shift and so forth, and humans appear to be more resistant to that. So people are interested. I have nothing other than that to say. Yeah.
So here next, yeah.
You mentioned the liver has mechanisms that the liver has mechanisms that preempt food ingestion, and so it's beneficial to eat on a schedule. Is that a recent adaptation? Because I imagine the hunter-gatherer ancestors might not have had food regularity. It was just as and when food was available, I guess I'm curious when that happened and how it worked for them.
So I don't have an answer to your question, but probably there was enough temporal entrainment just from daylight, from daytime. When you went hunter-gathering, right? When that took place, which also, you want to do that when the animals, when the lions are taking a siesta, et cetera, et cetera. There was enough. I'm making this up, right? But there's enough temporal association to probably lay the foundation for this kind of thing, even when your partner is not putting food on the table at 6:30. But that's, I don't have a real answer for you.
Down here in front.
Anthropologists talk a lot about what appears to be this biphasic sleep pattern that humans had until the 17th century or so, and they've said it's like maybe the nocturnal predators were most active on the savannah, in the middle of the night, and so that's why we woke up. But when you look at gene expression and glucocorticoid secretion, all these things, they seem to be on more like 12-hour
24-hour schedule, yeah.
So how do you think it was some cultural phenomenon that occurred for a few hundred years, or do you think that's real?
So of course, I don't know the answer, but my guess is yes. My guess is yes. And it's also possible that some of this is plastic, it's entrainable. In other words, if you do that for enough time or a few generations, that stuff changes, but your guess is as good as mine.
There's a mic up there. Yeah.
So, hi, Michael.
Oh, my goodness. Look at that. She worked in my lab. She's a UC graduate student.
Yes, first year.
Morgan Collins. Yep.
Great talk. While I was in your lab, I really enjoyed working with you and everybody else. But I definitely focus more on the molecular mechanisms, right, of circadian rhythms in general. So I'm just wondering if you could, speculate a little bit as to how you think the circadian rhythms change in humans as we age, right? So you see this different phenotype. And I'm wondering if you know or have any ideas as to what's going on molecularly.
With aging?
Yes.
So I don't know. There are people, of course, there are people who work on this because it's both interesting and fundable. My slightly cynical view on this is that everything gets worse with age. There are very few, except probably testosterone poisoning in the male population, which gets better with age. So I don't know if there's something very interesting and unique that could be boosted. I mean, the simple extrapolation from what I talked about is conceivably, eating on a regular schedule and keeping your sleep regular. It is likely to reinforce those systems which have gotten a little bit weaker, but giving you a more molecular explanation, a pill you can take, I don't know.
Question down in front.
Hi, I was just curious about what regulatory rhythms look like in species that are in perpetual darkness, like subterranean or deep ocean.
Right, so blind cave fish, for example, which people have worked on. So this is quite interesting. There's no light. Some species actually lose their circadian rhythms. It doesn't, use it or lose it. But the more interesting cases are the cave fish which have retained circadian rhythms. And so here's the explanation. It turns out the whole food chain in the ocean is under circadian control because, of course, there are small animals, plankton, et cetera, which are rising and sinking as a function of time of day. They're undergoing circadian metabolism, avoiding predators, et cetera. So this whole food chain is in the ocean. So the food is washing into the cave and out of the cave as a function of circadian time, despite the lack of illumination. And of course, this food entrainment system that I described is operating in the fish. So that's the interpretation of how they retain their rhythms. Jennifer, do I have it right? I want to ask her a question.
Down in front again.
So that's why I remember.
Great. Really appreciated your talk and like the parts about the brain and the liver and how important it is to keep them in sync. Are there other parts of the body where there's evidence that the circadian rhythm can become decoupled?
So that decoupling is a deliberate attempt to decouple, right? It's, in other words, it's normally coupled. And so it's the experiment which indicates what's the vehicle of coupling, which is, of course, nutrients from that. But I don't know of a natural decoupling except when you screw things up. And, of course, jet lag is one of those too, right? Which is, if you will, the human natural experiment for decoupling. But I don't know of a... I mean, the tissues are a little bit... I exaggerated slightly. They're not perfect. They're not sort of perfectly in sync, but that's not where you're going with the question.
So it's time for our coffee break. Please be back at 11:05, and thanks, Michael, for a wonderful talk.